Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amirault. I am the marketing manager at Medicinal Genomics and proud member of the team that puts on the CanMed conference. All right, just a few reminders before I introduce our guest for this episode. First, be sure to register for the CanMed 24 Innovation and Investment Summit before the early bird deadline of December 1st. That's only 1 month away, if you can believe it. We just announced last week that Golden Eagle Partners will be returning in 2024 with their Capital Markets Workshop that will cover topics related to investing in cannabis innovation. The session was a big hit last year with representatives from several top companies. It's a great opportunity to connect with them as well as potential investors. You can learn more at canmedevents.com and be sure to register today. We are also accepting abstract submissions for oral and poster presentations at CanMed 24. So if you have new or novel research related to cannabis science, medicine, cultivation, or safety testing, we want to hear from you. I put a link in the show description where you can learn more about the abstract submission process, and you can submit your abstract there as well. The deadline for that is November 18th, so get on it and good luck. Our guest today is Bill Graham. Bill is the founder of Microclone Tissue Culture, a company that provides tissue culture kits, consulting, and training to cannabis cultivators. With hands-on expertise, Bill retailed and distributed hydroponic supplies, sourced grow lights globally, created Biojuice nutrients, designed Phresh filter air quality systems, and simplified micropropagation cloning, creating Microclone tissue culture kits in 2005. During our conversation, we discussed the basics of tissue culture as a propagation alternative to cloning, how mother plants can lead to pathogen infections and loss of vigor, combining tissue culture and qPCR testing to eliminate pathogens such as latent viroid, and much more. Before we get to my conversation with Bill, I'd like to thank this episode's sponsor, Cambium Analytica. Cambium Analytica provides analytical, product development, and quality assurance services for the natural foods, botanical products, natural pharmaceutical, and dietary supplement industries. Cambium is a leading research organization that is focused on leveraging its expertise on drug discovery and method development to be a research partner of choice for new innovative companies in the burgeoning botanical medicine spaces. Their focus on applying analytical techniques in combination with sensory, product development, and consumer research is driving advancement in natural product quality and innovation. Learn more at cambiumanalytica.com. All right, and without any further ado, please enjoy my conversation with Bill Graham. Good afternoon, Bill. Thanks so much for joining us on the podcast.
Glad to be here.
All right, so let's talk about tissue culture. And I know this is a propagation method that you've been evangelizing for quite some time. And with the emergence of latent viroid and other plant pathogens, it seems like now more than ever, tissue culture is top of mind for many growers. So let's dive into it, and let's talk about what tissue culture is and what advantages that it may have over the cloning that a lot of growers are doing now. So let's start with that. So give us a high-level view of what is the tissue culture process, and we'll take it from there.
Happy to. Ordinarily, tissue culture is going to be practiced in 4 stages, and incidentally, I might just take this opportunity to kind of tell everybody what tissue culture is, especially as it pertains to plants. And very simply, it is hydroponics with sugar. So we're using the same nutrients. We've got some special amino acids. Usually there may be hormones to give it direction, but what makes tissue culture special from the hydroponics that growers are already doing is going to be adding sugar to give that energy source directly to the plant. Sugar can be directly absorbed by the stem and surface tissues without roots to provide that energy. So we can make plants from pieces that would be too small to clone very fast themselves. However, because it's sugar, everything else wants to grow on it. And that's when you start seeing the scalpels and forceps and the HEPA filter and the laminar hoods and the pressure cookers. All those things are to be able to make the growing medium, clean the plant, handle it, and grow it so that the fungus and bacteria and those things don't get back onto it. But given that, tissue culture is fantastic for being able to make lots of brand new clones from a sterile source. Once the plant's clean and it's growing in the jar. So the 4 usual steps of tissue culture are going to be introduction, multiplication, rooting in culture, and then rooting back out of culture. But you probably already know that I've kind of made a little special version of that to make it easier for growers and to make it more effective for a lot of our producers.
Right. So at a completely high level, what we're talking about is using small pieces of an established plant to make new copies of a plant. So not too dissimilar from what you would do with cloning, just with much smaller pieces, right?
Yeah. And in fact, it's funny. I'm actually calling it tissue cloning because we're using tissue culture to clone, and that's kind of part of my the shortcut that I've made up, and that is to top veg plants, taking a 2 or 3 centimeter cutting off of the top so that the plants will branch, which is valuable for indoor growth, especially. And this is primarily for our vertical growers, but works for anybody who is raising plants indoors in vegetative growth. But that's putting a tip cutting directly onto rooting medium, which is sort of a shortcut of the usual 4 stages.
Right. So you're taking, so a lot of growers, they're topping their plants, like you said, to kind of widen them and give them more of a bushy look. So you're actually being able to make use of that by putting it into a rooting media and kind of create copies. Now, would that not be a good material to use for cloning traditionally?
It actually makes a bad clone because there's so much concentrated branching in that piece. And to take it as a clone length, you're usually throwing away, 4, 6, even 8 good nodes, which is all growth that we don't want to get rid of. Typically, when a veg tray, which is usually about 96 or 100 plants, when it's growing, everybody in there is essentially a clone, but some are going to be more vigorous than others. And the technicians are going to look at it and say, okay, we have enough lower branching that we are going to remove this much material off the top of that plant and leave, is it going to be 4, 5, 6, sometimes 8 limbs, whatever their number is. But that piece has to go. And they're usually just pinching that to trash, pinching it compost. And so what we did is I was like, I'm in the facility already to do collection. They're always sending me to the mom plants to do it, but I prefer to go to the veg plants because immediately we can tell which ones are strongest just based on vigor. And number 2 is that it's already being done. So, this is material I can take that doesn't interfere with any other cloning or a propagation going on in the facility. And then it turns out that we could turn it into an actual strategy to produce all the plants that the grow needs, and in fact improve the crop quickly by selecting on vigor. And that's where things get really interesting.
Right. So, I mean, you touched on a good point there. So with your system, you could potentially do away with moms and just do all of your propagation off of your veg plants. Is that right?
Yes, I am mom plant's worst enemy. But I totally get it. What's really kind of funny is how we got here, because I, of course, ran the retail shops, made grow equipment. I and we used it. So I got to be the grower, equipment maker, and really mom plants were a means to an end. It got us by. As a grower myself, I never did mom plants because it was a waste of high intensity light.
Hmm.
I can get, if I'm producing all the veg plants I need for my grow, then I really just need like 2 or 3 flowering rooms, so that I'm producing veg plants every 2 or 3 weeks. So I've got all the fresh material I need to cut from. And when I was a college gardener, I was using side limbs for that work. Funny enough, that was exactly the same time I got into tissue culture, and it was going to be 20 something years later, I'm not dating myself, that I started realizing, wait a minute, this material that we're pruning off the top actually makes a great clone. And if we're taking it from the very best veg plants, then that vigor, which was expressed in the veg plant it was cut from, carries over into the new plant that culture is going to produce. And
Interesting.
we're able to get all the plants back over 2 pruning cycles. So if I've got a tray of 100 veg plants I need to replace, I can take about 35 of the best tops first, and then 7 days later go back for another 100 or so that we can be selective. And in 8 weeks, those tops going into culture were going to be the plants that are being topped again. So we're able to short the whole selection cycle down to 8 weeks. And you can see none of that involves a mom plant.
Hmm. All right. I'm not sure if I quite followed that. All the different stages
So
and the different multiplications. So
topping
try
the
to break
veg
that down a
plants to
little bit.
make the new plants that ultimately go back to veg.
Okay. So walk
I'm
me through
pulling
the
up slides while
process.
we're talking.
Say that
Go
again.
ahead. I'm pulling up slides while we're talking, so I can actually throw these up if we need them.
Oh yeah, absolutely. That would be great.
Got to
So
email them.
yeah, you could do that, or yeah,
I'm
if
just
you want to do that, I could pull it up. That'd be cool. Would it
okay.
kind of show the different
Yeah,
kind of
it's got a picture of the
different
whole thing.
multiplication steps there?
It does, but very simply, it just kind of shows a veg plant being topped, and then that going into a tube on rooting medium, which will grow about 3 or 4 times its original mass, because that's what's happening when this cutting is in the tissue culture tube. So if we've taken a 2 or 3 centimeter cut and put it onto rooting tissue culture media, the sugar is absorbed immediately right through the surface of the stem, and that cambium, that green growth tissue just underneath the surface, is going to be turning that into starch, complex carbohydrates. It's turning it into stored energy, wood. It turns it into wood, and you can see it get fat. Unfortunately, I took all of my last clones to Rhode Island with me, and didn't bring any back. I can run over and grab one from a show the other day, but it's got a little spoilage on it. But anyway, the bottom gets fat with all that stored wood, and it gets lumpy because of the influence of rooting hormone. So these shortcut tissue cuttings are ready to root into plugs in about 3 weeks.
Hmm.
So I'm using tissue culture as a cloning tool
So
without mother plants.
how many different copies can you get from one plant?
Well, if we were doing the straight micropropagation where one cutting would go into a jar and it would branch and grow on a multiplication hormone, that is going to be the class called cytokinins, which are the opposite of auxins, which are rooting. Those plants will usually grow to, if we're lucky enough to get them to branch, to like 6 or 7 new 1.5 centimeter node cuttings in the period of like 5 weeks. And that would make that many new plants. You start doing exponential multiplication on that, and you're going to have, we can have thousands. In the case of regular horticulture,
Yep.
I can tell you for a fact that a couple of plants can turn into tens of millions, because that's what I did with citrosa geraniums. If any of your watchers have the mosquito repelling geraniums in front of their house, and everybody recognizes them, those actually came from these hands in 1992, because that was my project when I was hired to work at AgriStarts, hired from college to return to college with a special 1-year project. And I did start with just 100 cuttings from the original stock plants. And those are the parents of all of the citrosa plants that anybody has ever grown in North America since 1992,
Wow.
which has to just be in the tens or even 100 million plants by now.
Wow. So are those all propagated through tissue culture?
They are now. Funny enough, the plants they sent me were in 8-inch pots from Holland, which is really interesting because this was a plant that was supposedly developed by somatic hybridization. The process was supposedly to take this scented geranium and grass of China, strip all the cell walls off, make the little naked stems stick together, regrow some cell walls, and then encourage those to grow in culture and become a geranium that had this citronella oil in it. However, it would, very much a tissue culture tool. However, the plants I got were in pots. And so that was when I first suspected that they were not actually from tissue culture. And the second was when I got a mosquito bite when I was in a greenhouse of like 200,000 plants. But I still encourage them because they're really pretty and they smell great, and I still have them all over my house. But that is tissue culture as far as a few plants making many. And that's where we really stand today as an industry. Most of the people who have are using your products, especially the HLVD testing, are testing plants that are going to be dedicated moms. And I totally get that. We try to encourage that to get away from moms and to try and do a lot of this work off of veg plants, for the reason that we kind of there's a disease cycle involved with mom plants that is unique to cannabis, indoor cannabis, and that is that the plants are the petri dishes for everything we're trying to get rid of, good or bad, and things are shared in innocuous ways. I mean, it's really hard to move the plants around and clean the tools every single handling, and most grows, are never going to be that disciplined. So anyway, even just air exposure in the same grow facility, everything we're worried about grows on cannabis, and the oldest plants are going to be the most exposed. And as your testing reveals so often, is that plants that show no outward symptoms can contain viroids, fusarium, botrytis, aspergillus, any of the things that we're really worried about. And of course, they are the source of all the new plants in the garden. And so the tissue culture shortcut helps break that, because the plants that will become the new crop will have to grow inside of a sterile tissue culture tube for at least 3 weeks, in which case, if there's anything there that doesn't belong, we can usually see it by eye. And of course, the medicinal genomics gives us tools for being able to PCR test that and know that the material in the tube itself is also is sterile, just in the course of the 3 weeks. But that's a good start. We can label it as a negative and then follow it afterwards and test it several more times. For anybody familiar with Segra or any of the companies doing tissue culture services to clean plants, they will be doing the exact same things, propagating the plants and testing over and over again. They usually look for at least 3 negatives from subcultures before they kind of give guaranteed viroid free back to the customer. And then it's, what does the customer do? They grow another mom plant, they start it all over again. They put mom in the same room as all the other mom plants. The technicians still have the dirty tools. Even if they're cleaning, there's risk. And we just want to kind of break all that. Besides, if I can turn a mom room into a flower room, I've just made a grower, a little bit more money,
Right.
sometimes 20%. And then if the crop gets better from just vigor, there's another 20, 30, 40% from doing nothing else except selecting on vigor, which incidentally is why we don't have corn that looks like this anymore. Because someone went in and said, hey, that one looks a little better. And that's what they grew next year.
No, and you make a great point about this, the moms being a bit of a petri dish or an incubator for all these different diseases. And it makes sense. They're older plants, and they're constantly being exposed to the air and the elements because they're continually having their limbs lopped off. So sort of breaking that cycle seems to make a lot of sense. And one of the things I had the pleasure of seeing you present at the Cannabis Science Conference just recently. And another thing that was brought up during that talk was this idea of genetic drift,
Yes.
which also seems to be sort of a problem that emerges from using mother plants. So speak a bit about that.
Oh, this is actually one of the things that got the kits popular the first time, is because there are a lot of fantastic growers, and especially legacy growers who have been growing plants for 10, 20 years or so. I mean, these are their trademark strains. But what's going to happen in that much time, especially if it's, recycled indoor growth, is it sometimes happens that the plants are just not yielding like they used to. They're not behaving like they used to. They're not yielding like they used to. But we know it's the same plant because you do the DNA testing of these things. Well, so, what that comes down to is epigenetics, which is going to be the expression of the genes. So they're there. Why aren't we seeing them? And why aren't they their very strongest? And there's usually a couple of things. They can usually be stress related. Because you've got a really nice indoor environment. The light comes on at the same time. The temperature is great. Everything is wonderful. If we lived in spas, I don't think we'd look quite as fit as if, we were outside, taking care of the land like our ancestors, right? Though it'd be worth it. But genetic drift is going to sort of just be like the plants sort of drop off of what they used to do. Tissue culture is a great little tool for that, because it kind of brings back juvenile growth. It tends to strip off some of the methylation that has occurred on that DNA in those nice environments. And frankly, we try to do a two-prong approach anyway, and that is for any grower that's experienced genetic drift, is to try to raise some of the plants vegetatively outdoors in the summer for like 6 weeks or so, for that multi-spectrum sunshine, the hot days, the cold nights. We can even simulate some pest attack with harpin protein and material like that. Possible real insect attack, wind, all those things like that are actually stimulation to the plant. We had several lectures during Cannabis Science about stress and so on like that. So I try to put the two together. And of course, we are going to take tip cuttings from those outdoor plants, put them into bleach water, which is sort of our conduit to getting plants into culture. And that is our quarantine to moving outdoor material back into the grow space. Plant cuttings go into the chlorine. We clean the tools every cut, which is how we are able to take that small piece of tissue and put it into a solution that's not only cleaning the surface, but preventing viroid or any other pathogens from moving between plants. And another one of the things we'll catch up on before we get out of here will sort of be the strategy of taking small pieces to hopefully hopefully take material without viroids and pathogens.
Yeah, so I mean, no time like the present. Let's get into that now.
Oh shoot! This is where we've been using your kits. This is where the qPCR has been. I beg your pardon. That was mostly for our chromosomes, but using your viroid detecting PCR has been excellent, because we're using such small material, and we can get smaller. I mean, 2 to 4 centimeters is a good working size for new users, because the plants that begin to root are a good size to go into a rooting plug and begin creating the new plants quickly, which is what growers want. But, as we teach in like our master class, is that, we'll go down to 1 centimeter, half centimeter pieces. We can keep going smaller and smaller, because this is the cool thing, is once you've got a simple setup, and I don't know if your viewers can see, what's behind me, but I've got a simple laminar hood, which is not even really necessary, and a lighted shelf with a HEPA filter. And this is my grow chamber. But we are able to, once we've got tissue culture media prepared, we can do plants of all sizes. But anyway, the premise, I have really just written into a proposal for a research paper is that the tips are always outgrowing viroids or anything systemic. So all the growth is coming from the very tip. I mean, less than a millimeter of the very tip is the future growth of that entire limb. And even before vascular tissues are produced. So the viroid and so on will move either in the vascular tissues or in the intercellular space between the cell walls and the cells themselves. Some will pass into the cells and some will be shared with the cells next to it, because that's kind of how cells communicate, but not quickly. It's all passive. And as long as that tip growth is vigorous, that passive exchange is not necessarily fast enough. Is there viroid in the cytoplasm in that brand new growth? And some of the work that Samir Punja, especially as we saw at CanMed, showed in his presentation is that viroids and systemic pathogens are not uniform through the plants. He had that one slide of a plant and he goes, the left side has viroid, the right side does not. Right. It was all the same plant. And where was I? My hand was sky high back there in the audience. I was like, let's talk about that, because that happens on individual limbs. And the more the plant grows limbs, the more likely that's the case. And especially the vigorous ones. So it kind of, one thing sort of led to another in all of that. But your stuff comes in because we can take a small cutting, and actually save a piece of the bridge tissue, which would be the stem tissue going into that cutting. If I take a 4-centimeter cutting, I can use the bottom centimeter for a viroid test, and the upper 3 centimeters are being cleaned to go to culture. And if we've got your tools for working on site at that location at the moment, we can know before that plant is even cultured if it was negative or positive on the test. So that's kind of an interesting one that I talked to, we talked about it in CanMed, and I talked to Kevin, is exploring the use of bridge tissue for PCR testing of viroid, because we shouldn't need much, especially now that I saw that you guys have tests with infinitely small amounts of sample. Like we can actually just stab the actual cutting with the tip of a micropipette. No cutting needed, no washing needed. So I'm really looking forward to getting into that, because we can now test the piece that's actually being isolated and grown to be the next crop. Negatives mean a lot more if we know that it's trapped inside of glass, and we're going to get a whole lot out of it.
Well, yeah, and so a lot of the talk about using tissue culture to sort of create a new copy that's free of HLVD had always been about meristem tissue culture, which I think when you were talking before is that very top tip, which you're hoping that the viroid hasn't had a chance to get to yet. But it sounded like you were just describing to take a larger cutting, but then test the bottom of it where it had met the larger plant to see if there's anything there. And if that's clean, you assume that the rest of the cutting is clean as well. Do I have that right?
Yes, and it is totally assuming. Yeah. And it's not necessarily 100%. We can't test every cutting, because if I need 100 veg plants and I'm doing 135 cuts to get it, that's a lot of expensive testing. But that's why it's called sampling. Mm-hmm. We will do some along the way. We got a whole strategy for this that kind of involves, weaning the viroid out of the crop gradually. But by gradually, it can be 20 or 30% a cycle, and those cycles can be 8 or 9 weeks apart. If you want to know a little more about that, But what we've gone on, because I do this for big growers, and I did it for myself. Interesting enough, by the way, everything that I came up with that we shared was developed for fusarium first. So when you all sent your first PCR test around with Kyle way back when, we were doing gender testing, but you asked right away, they were like, oh, what's the next test we would need? And I said fusarium, because at that moment I had a massive fusarium problem at my place. And so we did this really to eliminate fusarium, but the premise is exactly the same. And incidentally, it's turns out to be ideal for viroid as well, because they sort of tend to travel and move the same ways. But you know, if you're 100% trying to eliminate viroid in one shot, that's where we would take like those 0.5 millimeter meristems. And that can take a long time to grow a new plant, but you can. But what I discovered was that on a plant that we might have that might register positive, because it was positive because of where the tests were taken. It was taken at leaf whorls, you know, where everything, the pinnate leaves all come together, or down in the roots. And those are either vascular intersections where traffic seems to slow and so they accumulate, or of course the roots are a sink. You know, everything kind of goes down there and stays put. As a testing company, you want to find the greatest concentration of viroid so that you can get the result that, yes, this plant has viroid in it. But funny enough, I go to that exact same plant and do my stuff off of the tips, especially if it's been cloned and it's on the table and we've got vigorous growth. If we select on vigor, then, and this kind of goes down the type of research that Kevin's doing, is that we might have plants that are naturally resistant. We might have a strain of the viroid that is particularly weak. We might have any number of circumstances that just kind of mean that viroid is not necessarily a problem, not only in this particular crop, but in this particular tissue. If the tissue is vigorous, that's our sign. And you know, we've kind of assumed, just making up a number, is if we had a positive plant made into 100 clones, vegetative clones, and we start selecting on vigor on those, that taking tops, we're maybe taking off 20 or 30% that are absolutely viroid free, because the viroids will have not caught up with that tip growth. But 8 weeks later, those same plants are now our clone donors. So the plants from the test tubes that we topped are the veg plants being topped again, and we do the same selection on vigor. And now we take off another 20 or 30% viroid free. So if we start at a hypothetical 100%, we can go down to 80, 62, keep going like that. Pretty much at the end of a year, we're in the low single digits, getting smaller. And here's the thing, is most of the growers who have viroids, as your testing reveals, are living with it anyway. So we just try to strategically make that the very best growth that it can be. And besides, selecting on vigor seems to make the entire crop perform better anyway. Mom plants, as growers are using today, have been selected on vigor anyway, but that was 8 months ago. By doing it at 8 weeks, we can do that 6 times a year. You know, remember, if you're trying to do breeding experiments, you've got Arabidopsis for plants and fruit flies. You know, if you want insects, because we want those generations, you know, flip them. Right. Which is another culture trick, by the way, is we started doing, we started harvesting immature embryos, fertilized embryos at like 2 days. So we fertilize a bud, then dissect the flower and pull out the fertilized embryo, and can put that on culture as well. It's smaller than an eye. It's great, but you just put it right on the same medium, you know, a little less sugar, a little less no hormone, and give it time, and we can make plants from that. The value being is by just touching the tip of a scalpel to that little embryo, we can get our DNA sample for that entire cross. So then we can run it through PCR and be able to see it, is our marker pattern the desirable cross that we were looking for. Because that's how that goes, is you do a cross and your seeds still go all different ways. Right. And there's going to be a good deal of testing, but we can test the seeds for the genetics, and then from culture, turn those into whole plants after that selection.
Wow.
Yeah. All right. Now, this is where we work together so well.
Yeah. We veered off into a whole different.
I don't want to do what you're doing, but I want to keep sharing what we do. You know,
absolutely.
Walk them right up to the edge, and then you take them from there.
Excellent. No, and I want to go back to sort of this process of continuing to propagate to kind of get ahead of hop latent viroid. And so, as I understand it, so is the idea being that like Zamir had shown in his presentation, that the plant typically gets infected, it goes down to the roots, stays
Yes.
there for a little while, and then it kind of proliferates throughout the rest of the plant. So is the idea being that like you're trying to catch those tips before the viroid has had a chance to get there, and then you're kind of constantly
It actually
trying to
moves
outrun
up
it?
and down. So even if it's in those limbs, some of it moves down. The down tends to go down and stay down. Um, it could, of course, carry back up because the xylem, which will go back up to the limbs, is especially active during. It's active early on for water and nutrient salts. So of course it's the primary carrier for those. In flowering, especially in cannabis, it gets interesting because we have a new shunt, and that is that stored sugar, the wood and carbohydrates we're talking about in the roots and stems, begins to mobilize again. So it'll take complex carbohydrates and turn into the simple sugars again and start sending those up the xylem, which is when we suspect that the viroid is going to be carried up as well. Because sugar is relatively large organisms. It's obviously metabolically driven. And we suspect that viroid is involved somewhere in gumming up the metabolic process. But it's first of all, we're doing it in veg when those things would not be moving anyway. Number two is just that all that growth is coming from the tip. It's like trying to chase a rocket if you were in its burn stream. It's going to be hard. What was that very tip growth yesterday is now centimeters or inches higher tomorrow. That's incredible. And considering we're talking about things that are just shared passively, cell to cell, and when you get up into growth that high, and in those brand new vascular tissues, is they are often missed, is how it goes. And this is where the testing comes in.
Yeah.
Because, I've done this for a while, and we were sending in testing from tips, and we were getting so much more negative material. And what's funny is you guys, as a testing agency, were saying, oh, well, you should either mix all of those samples together, or let's go for these other high concentrated tissues, which is great, again, for identifying the presence of the viroid in the garden. I would almost go ahead and say it's there. And if it wasn't viroid, it's something else. But let's if we start doing it off of tips, there is the greater likelihood that, given the plant, assuming the plant had viroid in it at all, that we're using material that's so new and has grown so vigorously that it is viroid free. And if we take that cutting with clean tools, now the part about this I hadn't mentioned is that to take that top off, your hands have been cleaned in bleach water, your tools have been cleaned in bleach water. I actually have a vinyl apron with bleachy sponges in it and 3 sets of tools and gloves so that I can do that. And of course, from talking to Zamir, he said, yes, viroid can live in bleach as well, but not well and not for a long time. So it's the best we have.
Right.
Is to use that. We're not going to ask for trouble. We're going to do our best to eliminate it. But that will be cleaning the plant, cleaning the tools, and then taking another cutting. But that's it. If I can, I want to take my sample for the test from this piece on its way to culture. That's it.
Right. And so if I'm hearing you correctly, so if I'm a grower and I have a plant that tested positive for hop latent viroid, and maybe we're assuming that we were looking at one of those high concentration areas, maybe like the roots or one of those. So we know that there's viroid there. But I think from what I'm hearing is before you throw it away, you might want to search for a spot on that plant that doesn't have the viroid, take a cutting from that, and then try to use tissue culture to create a viroid-free copy.
Yes, thank you. And yeah, for sure. I would, and above that would be, taking like 100 or something copies and testing as many as you can afford, so you can have a representative sampling. Again, growers don't need to freak out because viroids in plants are shared by plant juice. It's only juice to juice that's going to make problems, which is why we clean tools. Think of it like HIV or hepatitis. You can touch the plants with your bare hands, You can move pots around and stuff in the room. When the leaves rub each other, it's not necessarily spreading. And we don't know so much about like the sucking insects, aphids, spider mites, so on like that. Those things should all be under control anyway, but um, the idea is that I would take like 100 and something cuttings and select off of vigor and do the testing on as many of those as you can afford. So we have a representative group.
Mm-hmm.
If out of 30 tests, we get 10 positives or 20, we'll keep the good 10 and the other 100 that we go, because you're still in business. These are the plants that made you tens of thousands of dollars just a week ago. We'll go ahead and grow them again, always keeping things clean between handlings. But then, we'll test the next round too, where hopefully we'll see, 12 or so positives. And then the next time, fewer than that. So, always a valuable part of it. And I would recommend, testing at least 20 of every 130 or 40 plant batch. So that you have a representative sample.
Right. And so this is a process you've used in the past and it's been successful?
Yeah. This is one of the best parts about it. It's why I call them buggy guys all the time, is kind of coming up and saying, hey, I'm doing this thing different, and strategically working out ways to make sure that we have a suitable sample, for the testing.
Excellent. No, we'd love to do an application note or a case study with some of your success stories. I think that would be a great thing to have on our website and share with our potential prospects too. So if that's something that you're interested in, we could do a nice co-branded document. I think that'd be awesome.
Okay, I'll send it to you as I go. I just put in another 800 words yesterday, so.
Oh, excellent.
It is. I will get the research done, because this is still, we've seen it on site, but science is science. There's a method, and we're going to do it properly. Besides, if I do enough of these, I may finally get that degree that I've been seeking for 30. It's crazy. I would have had a cannabis degree, if it existed. 169 credits, undergraduate, University of Florida, would not leave school. First, I was growing so I could pay for those credits, and they were only $99 each. So state schools, awesome. Everything was really kind of cannabis, horticulture, molecular biology related. It's just one thing right after another. I was like, oh, this is cool. They made me leave, and I tried to get a graduate degree, but no one was going to take me because they didn't know what I was doing. It's how it went. And I really just couldn't marry peas or beans or corn or something like that, to get in on with somebody's good graces.
Right. Yeah.
I'll talk to the ref about it.
We can't get you that degree, but we might be able to get you up on the CanMed stage if you publish that research, or if you send that research to us. So it's a decent consolation prize, I suppose.
I also promised to share a copy with Max Jones. Maybe we can wrap that all up. Everybody's in this game together.
All right, Bill, before I let you go, I want to give you a chance to share any resources that the listeners or the watchers might be interested in to learn more about your process and some of the research that you've done. And of course, plug away with any other ways that people can get in touch with you and learn more about what you're doing.
Oh, I definitely appreciate it. Thank you. I post all the graphics, links to the videos at microclone.com. We have the videos on YouTube under Microclone 100. Instagram is Microclone 100, I believe. Twitter, X, Microclone. Facebook, Microclone. It's Microclone everything. And I try to post little bits and pieces everywhere. So you can sort of build the story, but the website is the very best direct resource, and my email is bill@microclone.com, and I'll put up resources such as my appendix, or the index of the research that we've followed, mostly Zamir Punja, some of your early stuff on viroids. And detection, distribution, and anything related to the shortcut propagation.
Excellent. Yeah, and before when I was getting ready to talk with you, I did watch some of the videos of the process, and You make it look very easy. Is it as easy as you make it look? Because I think maybe in my mind and maybe other people's minds too, like tissue culture seems like a very kind of daunting process that, you have to be super clean and super precise. But like I said, you make it look very easy.
Yeah, I mean, if you're going to be full on professional and you've got 100, 200,000 invested in it, we'll put up the doors and the pass keys and so on. But genuinely, the whole idea is to learn, start successfully, get all the way through, and we do this in that little lunchbox kit you've seen me carry. We have prepared medium in our starter process usually works with like an instant pot or a stovetop pressure cooker to sterilize media. Our transfer area is as simple as a plastic storage bin, and we work in a little shallow saucer. The plants are cleaned in chlorine water. Our choice chlorine is going to be sodium dichloro cyanurate, pool shock. Really, it's 99% dissociative chlorine. So it's free chlorine and it evaporates. So I love it because we can work in it without rinsing, and that's a big deal. And we can make it stronger, but we will clean the plants in this solution, and I'll actually put a puddle of it in the saucer, right, so that as we're dissecting the plants, nothing is getting on there. So that gives us a still air box. And then lastly will be some sort of a clear grow area where we can have lights on a shelf and a HEPA filter running nearby to keep the air clean over there. So the pressure cooker in the kitchen, a pH meter and some mixing tools, and a real simple transfer area. I've done this at trade shows. I do this in hotel rooms. I have generally sterilized media in the microwave right next to the waffle maker in the morning, and put plants in that medium before the afternoon. We might have a little more incidence of contamination because it's all brand new, but that's usually for learning. And the crazy part is we still will end up with like 50% clean plants, right, that the grower will transplanting into plugs 3 weeks later. That's the whole idea, is that tissue culture is a grower's tool they can use in the simplest area and can always expand. It's absolutely scalable. I've got growers who are up to over 1,000 clones a day who started with the same Instant Pot that I'm talking about.
Right. It's really just a way to get their feet wet and get them comfortable with it, and then you can scale up from there.
That's it. That's
excellent.
That's the idea. Yeah, is take the intimidation out of it. It's a grower's tool. We're growers. Growers innovate. Look at what the hell we did when growing was illegal. Look at what we made work. The grow lights you have today, you spoiled youngsters. You should have had it back in my day. Walk to school uphill both ways. Is that we made stuff from what we had. I was like street light, warehouse light, whatever you got. We took it apart, rebuilt it so that it became a grower's tool, and we had to make that stuff work because you were not necessarily going to the store to get the answers you need, and you were working in the middle of the night, so they weren't open anyway. And it's the tissue culture version of the same thing. But go to microclone.com. You can see everything that we use and get started for less than $300. Get you a whole kit. And my commercial people can usually get started for less than 1,000 and train several people. And I'm available for consulting by Zoom or on site.
Excellent, Bill. Always a pleasure talking with you. Seems like there's a lot of opportunities for us to be working together. So excited about that. Thanks again for taking the time, and can't wait to see you down in Marco Island for CanMed.
Yeah, I just got my discount code from Brandon.
All right. So
tomorrow, I'll see you next year.
All right. Get that ticket before they sell out.
Thanks, Ben.
All right.
And won't wait till the last minute like last time.
That's you and everybody else.
Just want to make you sweat. Have a great one, man. You say hey to everybody over there. I look forward to seeing y'all again.
All right, I will do.
Cheers. Night.
I hope you enjoyed my conversation with Bill Graham. Check out the links in the show description to learn more about the topics we discussed. And thanks again to this episode's sponsor, Cambium Analytica. Our next episode drops November 15th. That's 2 weeks from today. In the meantime, head over to canmedevents.com now to learn all about our CanMed 24 Innovation and Investment Summit, which is coming up next May. If you like what you see, and I'm sure that you will, Be sure to register before December 1st, which is the deadline for our early bird rate. Also, if you're interested in being a CanMed 24 presenter, you can submit your abstract. The deadline for that is November 18th. Also, I invite you to follow us on social media. We're on Facebook, Twitter, LinkedIn, and Instagram. Just search for CanMed Events. Also, we would appreciate it if you would rate, review, and subscribe. It really helps us reach more people. All right, that's it from us. Stay safe, stay healthy, and be sure to join us for the next CanMed Coffee Talk.